Thank you very much. So it's a great pleasure to be here, and it's even a great pleasure to share the stage with Jordan. It's always emotional for me because he has been our hero by starting this clinical trial without taking the risk, so many risks, without knowing if this would have any benefits. So thank you, Jordan. And so what I will do is I will start my, yes. So I will start my talk, and after he will also speak, and after we'll answer questions together. All right, so I always have to start by my disclosure. All right. So thank you, Dr. Zarinski, because this is, it was a very clear explanation of gene therapy, so it will make my talk much easier. So as you know, the gene involved in cystinosis is called CTNS, and in the case of cystinosis, so you have a faulty gene that results in the accumulation of cystine. And so the treatment that are currently available for cystinosis are first supportive. We have to treat all the complications, but also we have really cysteamine change the course of a disease, fortunately. But they are treating the aspect of after the fact. So in gene therapy, what we are trying to do is really going to the origin of the disease and trying to treat the gene itself. But when I started, I wanted to do gene therapy, and when I started to work on cystinosis, we were looking for the gene. And I really wanted to go, because I wanted to do gene therapy, but at the time of my PhD, it was way too early and too science fiction to be able to do a PhD on cystinosis. So my mind was like, okay, I want to go from the gene to gene therapy. So I was like very determined to go from the gene to gene therapy. But the issue with cystinosis, and I didn't know that at the time, is that the gene CTNS is expressed in every single tissues. So that's challenge number one when you want to do gene therapy. Challenge number two is that the protein involved is a non-secreted protein. So that was the worst disease model for gene therapy, but I didn't know that. But, I decided to go up to the gene therapy, so I continued either way. And I was thinking, so what do we do when we want to bring the gene in every single tissues? So there is a vehicle, a natural vehicle that we all have in our body, and that's the adult bone marrow stem cells. And these cells can just travel everywhere in your body naturally if there is any injury. And so we tried, I tried every single bone marrow stem cells, the whole bone marrow, the hematopoietic stem cells, mesenchymal stem cells, and very surprisingly, the hematopoietic stem cells were the cells that were doing the best job. And I say very surprisingly because they are making blood cells. So it was very not obvious that these cells would actually do the job and try and have a beneficial impact on cystinosis. And so actually we have now the explanation, and I will just tell you a little bit after that. So what the approach that I'm using, as Dr. Zarinski said, is called ex vivo gene therapy. So we are taking the hematopoietic stem cells from the bloodstream of the patients, and we ex vivo modify them. So we take them to a lab, and we put them in the presence of virus vector to introduce the gene, or we can also gene correct them using CRISPR-Cas9. So in the case of cystinosis, we are using a virus vector, as I will say in a little bit. Then, after, you have to remove the bone marrow stem cells that are non-corrected from the body. So this is when we have to do a chemotherapy. the chemotherapy will really remove the bone marrow stem cells. So then when we do a blood infusion of a new stem cells, they will go by themselves, taking the space in the bone marrow. And once they are here, they will be there for the rest of the life of the patients. And so that's the good thing. It's a one-time treatment. And so this approach is well known for all the blood disease, like, immunodeficiency disease, because again, the hematopoietic stem cells are making blood cells. So it's very obvious that this can be used. But there is another type of cells that are called monocytes in the blood, in these blood cells. And these monocytes have the ability to migrate and go to the tissue and differentiate into macrophages or macroglia cells in the CNS, in the brain. And so these cells will go into and reside in the tissues, and they are able to bring the healthy protein into all the tissues. And so this is why we know that the hematopoietic stem cells were able to have a beneficial impact of on cystinosis. And so to correct or gene modify the stem cells in the case of cystinosis, we are using what we call the lentivirus vector, which is a healthy version of the HIV. And so we added in this vector the CTNS gene that is faulty in the cystinosis patient. And so now the new stem cells will have a functional CTNS gene. Then we transplant the new cells. And so we started, we did all the preclinical to clinical. We did, as Dr. Zarinsky explained, went from the bench to bedside. But today I will just talk about the clinical trial. So we started the clinical trial in 2019. And so the principal investigator are myself, Dr. Bruce Barshop, who are doing all the cystine measurement at UCSD and nationally. And we have Dr. Ball, who is the director of the bone marrow transplant. So the number of patients we were targeting are 6, because in the field of gene therapy, the number of patients for the phase 1/2 clinical trial are very small. And so the primary endpoint of this phase 1/2 are the safety. So this is what we want to make sure this is safe. And that's why we enroll only adults first. And we had several secondary endpoints to try to show efficacy. And so the patient had to stop cysteamine about 2 weeks prior to the chemotherapy, and the cysteamine eye drop a month post stem cell infusion. And so what happened when the patient enrolled? So first we do a baseline study. So we measure all the many complications. We really determine the health status of a patient before stem cell transplantation. Then what we do is that we do a GCSF mobilization, so that will take your stem cells into your blood, and you do what we call an apheresis. So we take the white blood cells from the body. This will be impacted in a bag. So we have a bag of stem cells that we keep at UCSD that are unmodified. In case something goes wrong with the modified stem cells, then we can remove them and put the normal stem cells as a backup in case something goes wrong. But otherwise, there is another bag that was transported to UCLA in what we call a good manufacturing practice room, where they can engineer the hematopoietic stem cells that can be transplanted into the patient. So here they take the CD34 hematopoietic stem cells, they put them with a virus vector called the lentivirus vector, and at the end you have like a mixed population of cells that will have the virus vector and some that won't. And that's something that is really patient dependent. We cannot predict beforehand how much of your stem cells will be gene modified. Then we cryopreserve the product. We do a lot of safety study on the product. And if it meets the release criteria, then it's bring back to UCSD. And then the patient is called back to UCSD. We do then the 4 days chemotherapy. And then once we have the chemotherapy done, the product is infused on the day after. And so I'm happy to report that now the clinical trial has been fully enrolled. So we have done the 6 patients out of 6 for this phase 1/2. So my job is done. Next, it will be our industry partner that will take over for the next phase. And so we treated 6 patients, 4 males and 1 female. The age range at the time of stem cell infusion was between 20 to 46 years old. So we had no adverse event or serious adverse event related to the drug products, to the same stem cells, so this is very good. And actually, we didn't get any serious adverse events, so this is really good. Most of the adverse event was mild to moderate, and mostly related to the stem cell mobilization, the chemotherapy, or the underlying disease. So when we do the chemotherapy, after about 7 to 10 days, the stem cells really drop. That's where it's risky, and that's why when the patient has to stay in the hospital, because then you have no immune defense. Then it takes about, I would say, 7 to 10 days to come back to normal. And so you have to stay about 2 to 3 weeks in the hospital, and then you can go out of the hospital because now your immune defense are back. And you can see here, I don't have a pointer, but we need the image, yes. I don't have a pointer, so it would be a little bit complicated, but you have a recovery of the stem cells of all the patients here. And you have a normal range for all of them. And for most of them, after like 3 to 6 months, everybody was back to normal. But we had 2 patients who had a little bit of delay. We had the patient 4 in blue line, who was a little bit delayed because he had COVID, but he was, after less than a week, he was back to normal. But we had, unfortunately, the patient 5 that I wanted to discuss about. The patient 5 actually had COVID like about a month after the stem cell transplant, and a very bad COVID, and she relapsed after, so she had more than a month of infection. And so at the time, we were scared about COVID because she was immunodepressed. So we thought it was risky. But what we didn't know, and what we still, realized, is that she still didn't recover normal white blood cells. And we learned after that actually COVID is attacking the stem cells, the hematopoietic stem cells, and especially the monocytes, the cells that I told you are making the job. Because you can see also below that her monocytes were below normal, and actually it was fluctuating a lot, and actually it was mostly sometimes she was at 0 monocytes. So meaning that there was no stem cells that could go into the tissues and help for the tissue recovery. So this patient, we had to put this patient back to reduced dose of cysteamine until the white blood cells will be fully recovered. So the message for the next phase is that really protect yourself. If you go to the clinical trial, protect yourself to COVID, not only because it's dangerous because you have immunodeficiency after bone marrow transplant, but because COVID will be very detrimental for your stem cells. So be careful of that. So to measure the efficiency of a gene modification, we measure what we call the vector copy number, which is the number of vectors per cell. And so you can see here on the right, on the left, the vector copy number of a drug product. And so the vector copy number goes from 0.6 to 2.9, so for patient 4, to 2.9 for patient 6. And after we need to see what's the fate of these gene-modified cells with time. So we take the white blood cells of a patient at different time points and measure this vector copy number. And so you can see, for example, for patients, so at 6 months, we usually have a decrease of a vector copy number. This is true for cystinosis, but it's true for most of the ex vivo gene therapy. And why? It's because the progenitor cells die, and the really true stem cells take over, and this is the one who will stay for the life of the patients. So you have a little bit of fluctuation at 6 months, and usually it's a decrease. Here, if you look at in pink, you have patient 1, and we were surprised to see, a very big decrease of vector copy number at 6 months. But the good news is that after, we saw a progressive increase of this vector copy number, and now patient 1 is stable at 1, so 1 vector copy per cell, which I think is, the body says, oh, this is pretty good now. Keep it that way. So I think it's a good thing. We have, otherwise, we have also several patients with a vector copy number above 1, and the highest is actually patient 6, who didn't even have a decrease of the vector copy number at 6 months. Is still at 2.5. So as you know, we follow up the patient by measuring the leukocyte cystine. And so we did that in our patients, and in all of them, we saw a decrease of the cystine content, despite the fact that they are off cysteamine. And so that's good, but expected too. But what we wanted to make sure is that the stem cells were also migrating to the tissue and doing their job by removing the cystine in the tissues, cystine crystals. And so we do skin biopsy once a year and rectal biopsy done by Dr. Ranjan Dohil, who is present here, every 6 months. So like that, we can measure the cystine crystals by a method that he developed. And we saw that there was a decrease and stabilization of the number of cystine crystals into the tissues. But for the long-term follow-up of a patient, we don't want to keep doing all these biopsies. So what we developed in my lab is a new method to visualize and quantify the cystine crystals in the skin using a confocal microscope. And so this is non-invasive. We just put a microscope here behind your ear, and we take a 3D imaging of always the same spot. And the cystine crystals here can be seen green. And we can, we're using imaging system, quantify very precisely the number of crystals. And here you see data from patient 1. So most of the data is from patient 1 because we have more than 3 years of data for this patient. And you can see, for example, at baseline, the number of green points, and even visually, you can see them decrease with time. But we have also, I really like, I think, this method, and I think it's very sensitive because what we've seen in all our patients is that at the time of a transplant, we actually see an increase of the cystine crystals, if you see the curve. And that's expected because the patients are off cysteamine, and the stem cells take about 2 to 3 months to really engraft into their tissue and do their job. So we can see that with the confocal microscope. But the good news is that after this 3-6 months time point, then we saw a decrease, showing that the cells start to really do their job. And actually for the patient 2 and patient 1, for whom we have the most data, you see that now the quantity of crystals are actually below 1,000. We have the data for the healthy patients because as control, and we have a background, and we are all between 500 and 700. So to be below 1,000 is a pretty good number. So we use the same approach using a confocal to measure and visualize the cystine crystals in the cornea. And again, we see data from patient 1. You can see the cystine crystals at baseline. And despite the fact that it was an off-cysteamine eye drop, we could see that the cystine crystals in the cornea were reduced and, stabilized at a point. And this was scored by Dr. Lian in France, who is expert in the cystine crystal. So that was not quantified by us. And the photophobia for this patient and for most of our patients are stable or even decrease after the stem cell transplant, like we can see here for patient 1. So what about the kidney function? So we have only 2 patients that didn't have a kidney transplant at the time of a stem cell infusion. It was patient 1 and patient 5. So what we saw is that both patients were in stage 3 chronic kidney disease at the time of stem cell infusion. So this is a too advanced stage for the stem cells to be able to rescue the kidney. So we hope for the next phase that the FDA will be able to approve for the stem cell to be done earlier to really determine the impact of the stem cells on the kidney function. But so then for patient 1, as you can see, the kidney function after kind of stabilizing for some time, decrease over time, and after 2 years post-stem cell infusion, he got a kidney transplant, and now his kidney function is back to normal. But the good news is, because I remember that before doing that, the FDA was a little bit nervous of what about if a stem cell does something bad to the kidney when the patient got a kidney transplant. But the good news is that what we've seen is that actually the kidney function of a patient who got a kidney transplant is the same than before or even better. So this doesn't harm in any way the kidney transplant. So that's the good news. So there was something that we saw with Jordan, and you will be able to see him after. Most of the patients with cystinosis, a lot of them are blonde with pale skin. And this is because, and it was shown that actually cystinosin is not only a cystine transporter, but is also involved in the melanin biogenesis. So that was making the skin and hair color. And we saw something, it's normal after a stem cell transplant and chemotherapy to have hair that become darker. But usually it goes away after less than a year, you should be back to normal. But actually what we were seeing with our patient 1 is that it's still not back to normal. It's still darker. So we're like wondering, maybe this is something we have to study because maybe it suggests that cystinosin is back and do this job with this skin color and hair color. So, so we decided to measure very precisely the hair color, and this is patient 3, actually, that when we started at baseline. And you can see that, so lower, and this means darker. So we can see that even after 2 years, his hair color keep darkening. So I think this is a very good sign. And actually now we are back to the mice, and I'm doing single cell analysis, etc. A lot of things to try to understand and see what's going on here, because it's very interesting. So now we have most of our patients, except patient 5, are still off cysteamine and cysteamine eye drop. And for patient 1 is over 3 years now. And most of the data, because I cannot show you all the data we are doing, but we are really looking at everything, looks very stable, so which is really good, despite the fact that we are off cysteamine. So I want to thank all our patients that you can see, in the blue circle or square. This is all our patients that have participated to this clinical trial. Again, they are the pioneer of this study. I want to do a big thank you to the family and their support, to the whole community of cystinosis, because this was not easy to get approved, or and we had so much support that the FDA was very supportive on this study. I want to also say a big thank you to all the people to my lab. I mean, it really takes a village to go from bench to bedside, and so they did an amazing job. I want to say also a big thank you to the Cystinosis Stem Cell and Gene Therapy Consortium, all the physicians. We have Ranjan Dohil, who is here. Evan Pont-Grimm has been a great consultant on all the study. We have Dr. Troner, also, who is here and participate to this clinical trial. I want to thank all the personnel and medical personnel, all our collaborators. I want to say also a big thank you to our industry partner, who was AvroBio. They really help us, on behind the scene. Now, you probably all know that this has been acquired by Novartis now, so they are the new industry partner, and I'm sure they will do a great job. But if you have questions on the next phase, they will be the one to go to. And so I want also to thank all the grant funded, because all this work has been funded by academic grants. So we have grants from the Cystinosis Research Foundation, the California Institute of Regenerative Medicine, and NIH. And thank you all for your attention. Thank you. So now I want to call Jordan. Yes, here. So, and I will answer the question with Jordan right after.
Hello, I'm Jordan. I went out last night, so I'm a little tired, so bear with me. But I'll just tell a little bit about myself, I guess. I was diagnosed at 8 months of age. When I was diagnosed, I was actually in chronic kidney failure. I was at like 20-some%. And I know it sounds crazy, but my mom fed me all like fruits and vegetables in the blender for a couple years, and my kidney function went up to 80-some%. So I lived with that for about 20 years. I was just like all you guys, dealt with the nausea, the pills smelling, hard time swallowing them, all that kind of stuff. I did a trial in Chicago for 7 years for the RP103. So after I did that till I was about 17, and then in 2019 I had the stem cell transplant on October 7th, and my kidney function was slowly declining after that. I went in with a little bit over 40-some%. And a year ago, I got my brother's kidney, and I feel pretty amazing now. I honestly have never felt better. Yeah, I guess, is there anything else you guys would want to know?
Okay, thank you, Jordan. So we can take questions. Yes, he deserves that. So thank you for sharing that, Jordan. Any questions from the audience? And I can get to you with the mic if anybody has a question they'd like to ask. Raise your hand. I see Katie. 1 second, Katie.
Jordan, this question probably is not for you, but more for Dr. Cherqui. Dr. Cherqui, can you address, oh, there you are. Could you address the risks of reproduction for women going through your trial and what you recommend?
Yes. So actually I can address this for both men and women. So the chemotherapy has the risk of not be able to get a kid after. You can be sterile after the procedure of chemotherapy. So the Cystinosis Research Foundation is funding the harvest of eggs and sperm for the patient to enter the clinical trial. So many of our patients got that done. So if they want a kid after, they still can have a kid. And I really advise to do that for sure before this procedure.
Thank you.
Jordan, how does it feel in your mind to be patient one? And as you move forward in communicating your current state of care and quality of life, what recommendations to the people in the audience who are looking forward to the opportunities in the future, how should they prepare?
Yeah, being patient one was honestly pretty scary. Like nobody really knew what was going to happen after I got the new stem cell transplant. It was definitely life-changing. I know, like, after I started feeling better and feeling good, I went out more with friends, and I think my mom was kind of upset at me. It was a whole new way of life. Like, I actually was not tired. Like, usually I'd hit 10, 11 o'clock or something, and I wouldn't go out. I'd just be exhausted, and then I would have all those pills to swallow, and I'd be nauseous, right? But it's definitely hard to get used to feeling normal. It's life-changing.
Hold on, I'll get over to your side. Excuse me, sorry. Big room. There's so many people here. It's really good. Getting my steps in. Can I scooch by you? So sorry. Galina.
Thank you both, Jordan and Stephanie. Question for Jordan. Jordan, with any gene therapy clinical trials, it's like, I think, 10 to 15 years follow-up, long-term follow-up. Just simple question, is it a burden? How difficult, to go through all of the follow-up visit when you feel good? You probably like, why do I need to go to do blood work and stuff? So just question about the burden of long-term follow-up for gene therapy.
So you're just wondering about how the gene
Yes.
therapy was?
Yes. Yeah. No,
Yeah.
like long-term follow-up, because you're still in clinical trial.
Yeah, coming back every 6 months, like traveling there and stuff wasn't bad. It was the testing. There was a lot of tests, like eye tests, and the blood work wasn't bad. There was 24-hour urine. I'd say, for me, the most annoying thing was for sure the eye test, because I had sensitive eyes. And the chemotherapy for sure was definitely the hardest thing. But coming back every 6 months, it wasn't really a bad thing at all. Now I go back once a year, so it's definitely getting better for sure.
And after 2 years, the follow-up are much lighter. So yeah, we have a lot of study during the 2 years, and after it's much less.
Jordan, Daniel Grant here from Novartis. I'm over here on. So really exciting to see Stephanie's story and hear from you. Having a stem cell transplant is a big deal, and I'm sure you had a lot of discussions about risk before making the decision to be part of the study. Can you tell us a little bit about how you thought about that decision and what it was like for you to ultimately make the decision to move forward?
Yeah, so I did the 3 weeks of testing prior to deciding if I was healthy enough or not to do the test. And then we went home for a few months, and once they said I was able to come back for a stem cell transplant if I decided to, I think I thought about it for, honestly, a month, and I sat down with my family, and we all talked about it and the risks and rewards, and we kind of explained it like there's, a tornado at the back door and like another storm at the front door, and it was hard to choose, because if I didn't do it, either way I was going to need a kidney transplant. Who knows what happens in the future? Or I do it, and if it doesn't work, from what I understood, I just go back on my pills, and at least I tried. So to me, I had to try and do something to better my life, and it was definitely hard because I had a full-time job and a house, but my company was pretty good. They actually gave me my wage so I could pay for my house while I was gone. So that was pretty awesome of them. And I had a lot of support. Honestly, if I didn't have the support from the cystinosis community and my family, it would have been mentally tough for sure.
I think we have time for one more question, Jordan.
As an adult with cystinosis myself, what do you recommend to step up and give us the strength to actually want to step in and do the trial? And if the second part of the trial is coming up, where do we sign up?
You see the gentleman behind you? You'd ask him. Yeah, no, I mean, so we have to realize that I have to say something. It's like they are a transition. Because it was AvroBio, now they have to learn, and that's why it's great that they are here to learn about the cystinosis and everything that goes with it. So it will take a little bit of time before they start the next phase. That's normal. But at the same time, it's a very good company, so I'm really sure that, they will do everything that they can to give bring that to all the patients.
Okay, thank you to Jordan. Thank you to Stephanie. Really appreciate your work.